Okay, so in our last class, so we discussed about peptide bond cleavage. Okay, so peptide bond cleavage, also acidic hydrolysis and basic hydrolysis. Acidic hydrolysis on cleavage of peptide by using acidic condition, there will be a breaking of all the peptide bonds, and since it is a acidic one, certain amino acids destroyed. And in the case of uh, basic hydrolysis also, there will be a bond breakage, peptide bond breakage. In addition to that, uh, racemization is the problem. So that means if you are starting with the L amino acid, you may end up with the D amino acid. So normally L amino acids are physiologically active. So that is the reason specific methods are required. In that specific methods, we discussed about hypobromide reagent. Hypobromide selectively cleave the peptide bond between the tyrosine and adjacent amino acid. Similarly, we discussed about sodium NBS. NBS means N-bromosuccinamide. So N-bromosuccinamide selectively cleaves the peptide bond between the tryptophan and adjacent amino acid. Similarly, we discussed about the what is called as uh, cyanogen bromide. So cyanogen bromide selectively cleaves the peptide bond between the methionine and adjacent amino acid. So we also discussed about the synthesis, how one can synthesize a peptide. So during the synthesis, it is observed that unwanted product formation. So unwanted product formation because of the interaction of amino group of the uh, uh, amino and carboxylic group of the same amino acid. In order to make amino group interaction of the amino group with the carboxylic group of the other amino acid, one should protect the amino group. So, after protecting, it is possible to get a desired product. So, in that, we also discussed about the key steps involved in a peptide synthesis. So, what are the key steps involved in a peptide synthesis? So, one is blocking, another one is activation, and one more is coupling, and one more is deprotection. So that means four steps are involved in a peptide bond formation. So look here, this is the first step. First step is here amino is protecting. Z is a protecting group. So then what is protecting group? Protecting group is the one which protect the amino or carboxylic acid, but it it should be inert during the synthesis, and it can be easily removed uh, without affecting the peptide bond linkage. So this is the it's called the protecting group. So here anyway, of course, maybe after few slides, one or two slides, we will go for a protecting group itself. What are the protecting group? More particularly, amino protecting group. We will discuss one by one. We are discussing nearly three amino protecting groups. So before that, you should aware about the peptide synthesis. The peptide synthesis first step is the protection of amino group. Now the obtained N-protected amino acid is activated by treating with thionyl chloride. So activation means it's nothing but a conversion of carboxylic acid to acid chloride. Why we want to convert? Because in this the OH group is not a better living group, whereas the chloride is a better living group. So once you convert into a chloride, this can be treated with the unprotected amino acid. So this one is protected amino acid, this one is unprotected amino acid. So that means this is acid chloride or activated amino acid made to react with the protected amino acid. This is a unprotected amino acid. So what will happen? There will be a reaction between the amino group of the one amino acid and acid group of the another amino acid. So there will be a linkage between this carbon and this carbon, forming a N-protected dipeptide. So this is, suppose here R1 is equal to H, then it is a glycine. If R2 is equal to CH3, then it is glycyl alanyl peptide. Suppose if you want to prepare a tripeptide, so what you can do after getting a dipeptide one, so then go for activation. So this carbon activation, and then treat with the third amino acid, and it will form a bond, a tripeptide is obtained. Afterwards also, you can couple with one more amino acid, activation and coupling until getting a desired peptide. Once the desired peptide is obtained, then you can go for a deprotection. So the here deprotected, and there are deprotection method. I will also explain in a later stage. So the obtained one is dipeptide. Overall, you should aware that peptide synthesis means it involves blocking, activation, coupling, and deprotection. So these are the steps involved in peptide synthesis. So with this, I will go to the next slide. So I think one can ask, what is the advantages? What is the advantage of protecting amino group? So for that, what you can do, you can explain this one. So look here, suppose a amino group is not protected. So then that time what will happen if there is no protection to the amino group? See here I am taking a glycine. Glycine was treated with acid chloride. Glycine acid chloride is obtained. If there is no protection, then this will act as a nucleophile. Then this will react with the one more molecule of the glycine acid chloride. Then it leads to the formation of glycyl glycine unwanted peptide formation. So unwanted peptide is formed. So in order to avoid such type of reaction, a protective group is introduced. So that means before going for activation, N should be protected. Then afterwards, uh, a good protecting group must exhibit the following character. It should be easily introduced to the amino group without causing the racemization of an optically active amino acid. It should be removable under sufficiently mild condition. It should be removable under sufficiently mild conditions without affecting the peptide linkage and other groups. Other groups may be a sulfur group or thiol group or a phenyl group, whatever it is. So it must retain inert. So the protecting group should retain as a inert during the formation of peptide. So these are the three conditions for a protecting group. So what is a protecting group? Protecting group, it should be easily introduced to the amino group without causing the racemization of the optically active amino acid. It should be removable under sufficiently mild condition without affecting the peptide bond, and it must retain inert during the formation of peptide. So this is about protecting group and their advantage uh, uh, about protection of amino group. So with this, I will go to the protecting group, more particularly N protection, otherwise amino protection. Okay. So I will go to the next slide. So protection of amino group. So based on this, one can frame a question, write two methods of amino protection. So this may be a 5 marks question, or maybe a 4 marks question, or maybe a 6 marks question. The important amino means we can call it as a N protecting groups. So N protection or amino protections, the important ones are benzyl oxycarbonyl group. So benzyl oxycarbonyl group, it is represented as a Z. So Z means benzyl oxycarbonyl group. Tertiary butyl oxycarbonyl group. Tertiary butyl oxy. Here you can small t you can write here. So then it's called BOC. BOC means tertiary butyl oxy. Okay. B stands for butyl. O stands for oxy. C stands for carbonyl. Butyl oxy carbonyl. Normally butyl oxy means it should be a tertiary butyl oxy carbonyl. This is also a N protecting group or amino protecting group. And one more, fluorinyl methoxycarbonyl group. So fluorinyl is one moiety, just like benzene is there, no? So fluorinyl, I will tell you what is the fluorinyl group. Okay. Here fluorinyl group means, so in between two benzene, a 5-membered ring is fused. So fluorinyl is nothing but a benzene, 5-membered ring, and benzene. So two benzene, in between two benzene, one 5-membered ring is fused. That compound is called fluorine compound. But I will tell you what is fluorinyl methoxycarbonyl group. It's called F mark. So with this, we will go for one by one. So one by one means what is benzyl oxycarbonyl group? How the benzyl oxycarbonyl group is synthesized? How the benzyl oxy group is protected during the amino protection? And how the deprotection? Okay, those things we will discuss now. So that means we will discuss what is amino protecting group, how it is prepared, and after the preparation, how it is protected with amino acid, and after the protection, how it is deprotected. So those things we will discuss under these three molecules. The first one I will take up benzyl oxycarbonyl group. So this is the one, benzyl oxycarbonyl group. Oh, okay. Say this C6H5CH2 is called benzyl group. C6H5CH2 is called benzyl group. This is oxy, and this CO. This is nothing here, no bond is there. Don't write any bond here. So this group is called a Z group. Okay. So this method was introduced by Max Bergman and Leomide in 1932. This invention made modern peptide synthesis possible, and still benzyl oxycarbonyl chloride is the first choice of protection of amino group in the peptide synthesis. Then how this benzyl oxycarbonyl chloride or Z group is synthesized? I think you are aware about the alcohol. So name of this alcohol is called benzyl alcohol. Benzyl alcohol is treated with the POS gene. I think you should aware about the Bhopal disaster. Okay. 1 minute. Let's give an entry. Okay. So because of the Bhopal disaster, somewhere in the 1970. So because of that, even today, it's very difficult to grow a plant in that area. So what they used to do, they used to take a postin and they used to treat with amine. They used to make a ketene. That ketene was treated with, that ketene was stored in today, and the next day they used to couple with amine. So during that time, so the ketene was hygroscopic in nature. Okay, if there is a moisture present, it will react in a explosive manner. So that is the reason it's exploded in the Bhopal disaster. But here, of course, it is a liquid, okay? Just like methanol, ethanol is there, no? In that way also, this is a liquid, and it is stored in a tight, airtight bottle. So what will happen? The lone pair present on the oxygen, or the bond pair between the oxygen and hydrogen, there will be a new bond formation between oxygen and this carbonyl group. This Cl and H go out as a HCl. So if you want to do this reaction, what you can do, you can take ethanol in solvent like methylene dichloride or ethylene dichloride, and add a little amount of water and then sodium carbonate, and then add this phosgene to here. So that's all. So once your addition is completed, this you will get a benzyl oxycarbonyl chloride. Okay, that's about the formation of benzyl oxy. This is also a liquid, and this can be stored in a airtight bottle. So this benzyl oxycarbonyl chloride is obtained. Once the benzyl oxycarbonyl chloride is obtained, so it was treated with amino acid. So it's called protection using benzyl oxychloride. So what will happen? There will be a new bond formation between this nitrogen and this carbonyl group. Cl and H will go out as a HCl. So what is the product obtained? The product obtained, you can observe that up to this group is called benzyl oxycarbonyl. Benzyl oxycarbonyl amino acid. So with respect to the R, this amino acid changes. If R1 is equal to H, then it's a glycine. If R1 is equal to CH3, then it's alanine. If R1 is equal to CH2 phenyl, then it's phenylalanine. So whatever it's a general amino acid, and it's a general method of protection of benzyl oxycarbonyl chloride to the amino group. Now it's called Z amino acid. Z is nothing but a benzyl oxycarbonyl carbonyl group. So with this, I will go to the next slide. In the next slide, I will explain about deprotection. So maybe you know that now this is a Z amino acid. So this was subjected to activation and treated with a different amino acid to get a desired peptide. Once the desired peptide is obtained, then one should go for a deprotection of amino acid. So that I will discuss in the next slide. So deprotection, it's also called as a cleavage condition. The removal of benzyl oxycarbonyl group, Z group, from peptide. Removal of benzyl oxy group from peptide can be done in three ways. So one is catalytic hydrogenation. So catalytic hydrogenation means passing hydrogen through a suspension of palladium. Palladium is a catalyst. Okay, in a solution of uh, 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 Z okay, Z amino, what is called peptide, isn't it? So this is not required. Up to solution, okay, a bubbling hydrogen through a suspension of palladium. So in a solution of peptide, you can write that N-benzyl oxycarbonyl chloride. It's okay, you can write N-benzyl oxycarbonyl peptide. Yes, you can write, it's right only. Then what will happen? The hydrogen is there, no? That hydrogen cleaves the CO group and convert CO, releases CO and converts benzyl oxy to the mm, benzyl alcohol. And also, you know that one more thing also is happening here. So that means I will take up this slide. Look here. So H2, 1H will add to here. Okay. So this will go out as a carbon dioxide, and this will picks up the proton. So it means that 1H will add here, this will come over here, and this will picks up another H. So what you need to remember that when benzyl oxycarbonyl chloride is hydrogenated, 1H will add to this carbon. That means toluene is the one product, carbon dioxide is the another byproduct, toluene is a solvent, okay, and amino acid is the product. So that is about the catalytic hydrogenation. And it is also possible deprotection of Z group with hydrobromic acid. Hydrobromic acid in acetic acid. Acetic acid is a solvent. So what this is a process treatment of benzyl oxycarbonyl peptide with hydrobromic acid. Hydrobromic acid means HBr in acetic acid. That leads to the deprotection of Z group of the peptide. So Birch reduction, you know that Birch reduction. What is Birch reduction? Liquor ammonia, sodium in liquor ammonia is called Birch reduction. So sodium supplies electrons. Okay, electrons will go and sit on the CH2 group of the means benzyl group. So that later it will picks up a proton from ammonia. So overall, what is the byproduct? Benzyl oxide group is converted into toluene and carbon dioxide, and free amino acid or peptide is obtained. So these are the, so sometimes they may mark 2 marks questions on this. So give the deprotection conditions for a Z group. Otherwise, give the deprotection condition for benzyl oxycarbonyl group. Then you should be able to write all these things. So this is about the one amino protection that's called benzyl oxycarbonyl group. Okay, it's a preparation, it's protection, and it's deprotection. So with this, I will go to the next amino protecting group, and the next amino protecting group is called BOC. Okay, so benzyl. Okay. So here, by taking a benzyl oxycarbonyl chloride, I am explaining how a dipeptide is synthesized. So more particularly, glycyl alanine. So how the question can be framed on this? Give the synthesis of glycyl alanine. Give the synthesis of glycyl alanine by employing benzyl oxycarbonyl method. Okay, right. Otherwise, what we can say that this is a benzyl oxycarbonyl method. Otherwise, Bergman method is illustrated by taking the example of synthesis of glycyl alanine. So here the 4 steps are involved. Okay, maybe you can look into that. This is the benzyl oxycarbonyl chloride. So this was treated with amino acid. So here amino acid is glycine. Okay, so the lone pair or the bond pair react with this carbonyl carbon. So H and Cl will go out. There will be a formation of product, and this product is called Z amino acid. Otherwise, benzyl oxycarbonyl glycine. Benzyl oxycarbonyl glycine. Okay, so this was subjected to activation. Activation means treating with thionyl chloride. So what is the role of thionyl chloride? It converts acid into acid chloride. How it will convert? It will take up the OH present here. Okay, OH present here. Uh, Okay. Uh, no. What is happening actually? The H present here, no, that H go with one of the Cl, and one more oxygen, and this SO will go out as a SO2, and another Cl will attach COCl, uh, means another Cl form a bond with this carbon. So that is how acid chloride is obtained. So once the acid chloride is obtained, now it is activated. So this was treated with unprotected. Here there is no protection, you look here. But in this there is a protected amino acid. This is unprotected amino acid. So there will be a new bond formation between this nitrogen and this carbon. Cl and H. HCl will go out. And this reaction is called coupling reaction. And what is the product? The product obtained is Z peptide. More particularly, it is called Z glycine alanine. And then deprotection. So deprotection, more particularly, there is a breakage here. And here the conditions, you can use it as a HBr in acetic acid, or Birch reduction, sodium in liquor ammonia, or hydrogen in presence of catalyst. So what will happen? One hydrogen will add to here, so that is how toluene is obtained. Another hydrogen will add to here, that is how NH2 is obtained. And this CO2 will go out as a gas. CO2 is a gas, it will go out. And what is the product obtained? The product obtained is glycyl alanine. Okay, this is about how the a peptide, dipeptide is synthesized by employing a benzyl oxycarbonyl group as a protecting group. Okay, so this is about the benzyl oxycarbonyl group. So with this, I will go to the next protecting group, more particularly amino protecting group. So that is BOC. So BOC means tertiary butyl. Tertiary means CH3, CH3, CH3C. This group is called tertiary butyl group. Tertiary butyl oxycarbonyl. Tertiary butyl oxycarbonyl group is called BOC group. Okay, so what will happen here? Uh, uh, okay, uh, look here. So in the previous case, benzyl oxy chloride is used for the protection of amino group. But instead of chloride, here paranitrophenol is used. So because of the stability. Why? Because tertiary butyl oxy carbonyl chloride is unstable compared to the tertiary butyl oxy paranitrophenyl ester. Okay, so this one is more stable compared to the chloride of BOC. So that is the reason esters are used. So that means paranitrophenyl ester of BOC. So this was treated with amino acid. What will happen? There will be a new bond formation between this nitrogen and this carbon. And this will be the living group. So now the obtained is BOC amino acid. So this BOC amino acid subjected to activation here and treated with one more amino acid. The obtained one is dipeptide. So later one, and this process means activation coupling. Activation coupling can be carried out until getting a desired peptide. Once the desired peptide is obtained, so it can go for a deprotection. So in the next slide, I will explain how the uh, tertiary butyl oxycarbonyl group is deprotected. So look here, deprotection. So the removal of BOC group from N-tertiary butyl oxycarbonyl peptide can be done in two ways. So one is with the 2-normal hydrochloride, another with trifluoroacetic acid. So here I am explained with the mechanism itself. So now this is a Bach peptide. So this, when it is treated with H plus means more particularly 2 normal hydrochloric acid. Of course, trifluoroacetic acid also releases H plus. So what will happen? So you know that uh, 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 this bond pair will come over here. Why it is coming here? Because this bond pair is moving over. Why this is moving over? Because the lone pair or the bond pair is involved in a bond formation with H plus. So that leads to a cleavage. So cleavage between tertiary carbon of the tertiary butyl carbon and oxygen. Okay, so then the obtained one is carbamic acid. NCOOH is called carbamic acid. So this is also in presence of H plus, the lone pair present on the nitrogen picks up a proton. When the lone pair is involved in a bond formation, this bond pair cleaves. And this then when this bond pair cleaves, the bond between the oxygen and hydrogen cleaves, and giving rise to carbon dioxide will go out as a gas, and the obtained one is a peptide. So this is about what is called as Bach. Okay, so the use of Bach in peptide synthesis and how it is protected. It is protected with the help of phenyl paranitrophenyl ester. Once the desired peptide is obtained, it is deprotected by using a 2-normal HCl or trifluoroacetic acid. This is about what is called as Bach. I think you know that up to this benzyl oxycarbonyl chloride, butyl oxycarbonyl paranitrophenyl ester, both these groups are cleaved by using acidic condition. Acidic one is H plus condition. Of course, benzyl oxycarbonyl group, it is also cleaved by using Birch reduction or catalytic hydrogenation. But one more method is there, that's called FMAC. Okay, FMAC means fluorinyl methoxycarbonyl group. Okay, you can observe that this is one benzene, this is 5-membered ring, this is benzene. Okay, this group is called fluorine group, FL. Okay, here you can write pure, this is a fluorine, FLU. Okay, FLUORENE, fluorine, fluorine group. Okay, so fluorine, now it's called this is when we are naming this we will take it as a substituent, then we can call it as a fluorinyl methoxy. CH3O is called methoxy, and this CO is called carbonyl, and this group. So fluorinyl methoxy carbonyl group is called FMAC group. Okay, uh, and this one is used as a an amino protecting group. And this FMARC group, if need to deprotect it from the peptide, that time a mild basic condition is used. So because of this application, it is spread practical importance. So because in a mild basic condition, so we can remove the protection of FMARC group. So because of this deprotection conditions, so this protection is widely used. So now let's look into the how FMARC means FMARC chloride is synthesized. So this is fluorine, FLUORENA, fluorine. So this was treated with a base. Sodium hydride is a base. Ethyl chloroformate. Ethyl chloroformate will act as a source of uh, CH, source of Cl, CH2. So maybe what you can write, Cl, okay, and then CO, and then OET, Cl, CO, OET, okay, is called ethyl chloroformane. So then base is there that picks up this proton. Hydrogen is the living group, and there will be a formation of anion. So that anion react with the this formaldehyde. So there will be a bond between this carbon and this carbon. So forming a CH2OH. Once the CH2OH is obtained, so this was treated with phosgene. COCl2 means phosgene. The lone pair present on this oxygen attack this carbonyl group. Cl, H and Cl will go out, forming a F-mark chloride. So this is how FMA chloride is obtained. So the obtained FMA chloride is treated with amino acid. So this is a protection of FMA to the amino acid. So there will be a new bond formation between this nitrogen and this carbonyl group. Now the FMAC is obtained. FMAC amino acid is obtained. So the obtained FMAC amino acid is subjected to activation of this coupling. Activation coupling leads to the desired peptide. Once the desired peptide is obtained, it was subjected to deprotection of FMAC. So in the next slide, I will discuss about the deprotection of FMACs. So you know that this compound is called piperidine. So piperidine is a organic base. So just by using the piperidine, so we can deprotect the F mark group in a peptide. So this is the application about the uh, in peptide synthesis, because you know that amino acids are sensitive to acidic as well as the basic condition, but piperidine is a mild base. It does not affect the amino acids. So that is the reason this method is more popularized. So how this is cleavage occurs? The lone pair present on the piperidine, okay, the lone pair present on the piperidine can obstruct this proton. So what is the name of this proton? This carbon is alpha carbon, this carbon is a beta carbon. So then this proton is called beta proton. So what will happen? This bond pair is coming over here, and this bond pair will come over here, and this bond pair will picks up a proton. So CO is going out, and name of this compound is called dibenzene. This is one benzene, this is another benzene. Dibenzene furene, Furene, furene now. So is the byproduct. This is a byproduct, and carbon dioxide is another byproduct, and it's a peptide is the product. This is about the amino group protection. So today we discussed about amino group protection and its importance. More particularly, we discussed about benzyl oxycarbonyl group, how it is protected, how it is synthesized, how it is deprotected. We also discussed about what is called butyl oxy, tertiary butyl oxycarbonyl chloride, uh, how it is protected, how it is deprotected. We also discussed about FMAC group, okay, how it is synthesized, how it is protected, how it is deprotected. Okay, this is about today's class. Maybe if you have a doubt, you can ask. Otherwise, you can log out. Maybe if you have a doubt, you can ask.